The future of Caribbean reefs is also closely tied to the ability of corals to successfully reproduce, while ecological conditions continue to change and degrade. Only in the past decade have scientists examined the details of coral reproduction. And in another study at the Smithsonian Tropical Research Institute in Bocas del Toro, Nancy Knowlton, a Scripps professor and director of the Center for Marine Biodiversity and Conservation, investigates how corals propagate in relation to environmental conditions.
Well, when we first started this work, we were trying to find out how many species of corals were really out there. And one of the best ways of distinguishing different species of corals is to see if they have different patterns of reproduction. So our initial studies had to do with determining whether, in fact, there were barriers to fertilization between the three species that are in this closely related complex of corals. Now we've moved on to looking at fertilization biology in a more general sense, trying to see when successful fertilization takes place and when reproduction is unsuccessful. And that's a big part of what we're doing right now. So every year we come out here and monitor the percentage of eggs that get fertilized under different circumstances.
Joining Nancy Knowlton in her research is Professor Don Leviton, a biologist from Florida State University, who is an expert on marine fertilization. They've been working together in the Caribbean for about a decade. Each year, they come to Panama with a group of students to monitor an amazing event, the mass spawning of corals. During these events, they collect data on the timing of the spawning, the rates of fertilization, and the distances traveled by the eggs. They also collect samples for genetic analysis. Nancy explains the procedures that will be performed while diving on the reefs. In the late afternoon, the research dive team assembles on the station's dock, where they prepare for the evening's work. Living samples of Monastrea annularis, or mountain star coral, are placed into buckets so that they can be taken onto the boats for experiments later in the evening. Once all the preparations are made, the team heads out to the dive site. The corals only spawn 1 or 2 nights each year in the late summer, when they release their eggs and sperm into the water in what is called a mass spawning. The team arrives at a site over a patch of healthy mountain star corals. In the hour before dark, they prepare for the dives. Green glow sticks are activated so that they can be used to light a pathway along the reef where star coral heads are located. The divers will follow this underwater survey line when it is dark. Past experience has revealed that one species begins spawning at 8 and the other 2 at about 10. So there will be 2 periods of intense diving. At about 7:30 p.m., the divers enter the water for the first spawning. 8 members of the team will dive, while 2 others operate a chase boat. The water is warm, but the divers wear full suits because of the long time they will be underwater. Normally, it's pitch dark underwater, and the divers work along the reef with just their flashlights. But tonight, the camera lights make it artificially bright. Large heads of mountain star coral line the reef. The water is somewhat murky because many types of plankton and small animals are active on the reef at night. The divers examine the condition of the polyps. When a coral is ready to reproduce, its surface becomes covered with small pink balls that contain bundles of eggs and sperm. When a colony is ready to release its bundles, a diver marks the coral with a lead-weighted glow stick with an identification number. The water becomes even murkier as hordes of small worms and crustaceans come out to feed on the nutrients in the water. Here you see the release. Those are not individual eggs, but instead are bundles of about 80 to 100 eggs glued together with sperm. Each polyp produces an egg-sperm bundle that is released into the water. These corals cannot self-fertilize, so the bundles float to the surface to mix with other bundles from other colonies. When the coral spawning is at full peak, a diver breaks the glow stick loose so that it can be used to follow a bundle to the surface. The bundles rise at a rate of about a foot a minute, which takes about 15 minutes at this site. You can see the surface is covered with floating egg-sperm bundles. The diver releases the glow stick and returns to the bottom to monitor another coral. At the surface, the glow stick is allowed to float around for about 15 minutes and is then scooped up into the chase boat. A plankton sample is taken to collect the sperm and eggs associated with the coral that the glow stick marks. They mix part of the sample with the eggs from the corals that were brought from the lab to see if the sperm in the water is capable of fertilizing them. They record the glow stick's number and take a GPS position to mark the site. The studies have shown that only those corals that reproduce near the same time as their neighbors are successful in fertilizing. Nancy and the other divers are excited about what they saw.
We got repeat spawning of things that we observed spawning last year, which means we can tell if there's year-to-year consistency in the time that individual colonies spawn. And we also got spawning records for one of the 3 species that previously we'd never gotten from this part of Panama, so that was great.
The team goes on to conduct a second round of dives later in the night. At about midnight, they return from the field. It's late, but their work this night is far from over. All of the samples are brought back to the lab for detailed analysis. Individual eggs can easily be seen in the cup. They are placed into a small maze that spreads them out into straight lines. The researchers can tell whether the egg is fertile because after about 3 hours, it is divided into multiple cells. A microscope is used to examine the slides and to score the samples for rates of fertilization. Here you can see some cells that have successfully divided. A counter is used in one hand to score fertilized eggs, while a counter in the other hand logs unsuccessful eggs. Tests are also made on the impacts of various salinity levels on fertilization, as well as the effects of several other environmental conditions. They found this year was an exceptional spawning with success rates of 80, even 90%, the kind of year corals depend on in order to propagate themselves from one generation to another. Yet Nancy Knowlton has great reservations about the coral's long-term future.
A recent study was published showing that about an average of 80% decline in the coverage by corals in the last 3 decades, which is really a very serious level of decline when you consider the fact that adult corals are typically 1 or more centuries old. So these mortality events are not something that reefs can recover from in the course of just a few years or even a few decades.